the cause.
Technology.
Anything else?
Technology.
Technology. So let's, okay, so three important, I believe, areas. The education, privacy issues. I would say these are the two most important. When we talk about education, what do you mean by education? If I say elaborate, what would you write? What do you mean about education? So education means educating pharmacists, physicians, and all other healthcare professionals. But educating the patients, right? Because as you may know, patients may not adhere to their drugs, right? So you do also have to convince them that it's good for them to take genetic testing. So you have to educate them about genetic testing and, pharmacogenomics. Talking about issues, what are the issues? And we talked about privacy. What are the other issues? The cost. What are the other issues? Policies to protect patients, right? And to protect healthcare professionals. Okay, so can you remember this for the midterm? Okay, great. So now, let's understand clearly what do we mean about pharmacogenomics and how you can use it. So in order for you to know how to use it, you need to know the drug response, right? And I'm sure you already covered pharmacokinetics and pharmacodynamics and pharmacology and therapeutics. So when you take a drug or a patient takes a drug, the drug has to go through the pharmacokinetic process, right? And through the pharmacodynamic processes. So what is the pharmacokinetic process?
What is that?
I can't hear you.
Admin?
Most your answers are correct, but you just have to speak louder. Okay, pharmacokinetics is what your body does with the drug. So what is it? If I tell you again, write something about pharmacokinetics and elaborate on every single process. What are the processes? Absorption, distribution, metabolism, excretion or elimination, right? So every time the drug moves through your body, it has to go through a different process and a different or friction, right? It has to cross membranes, right? So what happened to the drug during the pharmacokinetic process? What happened to a prodrug? Metabolized into an active metabolite, right? And then what? Metabolized by enzymes, right? Yes? Okay, so every time your drug moves from absorption, distribution, metabolism, it's going to cross membranes, right? It's going to cross barriers. So it may come across a transporter. It's going to be metabolized, right? It has to be excreted. So every single process involves enzymes or proteins that are helping the drug moving. If it's not passive diffusion, it's going to be active diffusion, it's going to need help, right? So when it crosses, every time it meets an enzyme, you're going to say, oh, I hope I'm going to be the lucky one. My enzyme is 100% wild type, 100% functional. Then the drug is metabolized the way the clinical trial has established should be metabolized, the way FDA has approved it to be metabolized. Meaning you can lose like some 80% or 30%, but whatever, stays in your body for a certain period of time will be the right concentration or the right dose to produce about a clinical or therapeutic effect, right? So what happens if the enzyme has a mutation? If one of the enzymes has a mutation, then the drug ADME that has been approved or published or established or clinical trials is no longer the same for some patients, right? Then for these patients, a different dose has to be calculated, maybe a different drug has to be prescribed. So genetic variation in drug metabolism and drug transporters is influenced by genetic variation or polymorphism, right? So how about pharmacodynamics? For example, you know that your drug can be classified as beta-adrenergic, alpha-adrenergic, or others. So your drug binds to a protein. How if that protein or receptor is mutated or variant, then you are losing the right drug response. So your pharmacodynamic pathway is also influenced by the genetics. So in general, drug response, because it includes the pharmacokinetics and pharmacodynamic processes, can be influenced if you have mutation in enzymes, proteins that are involved in the ADME or the drug response. So does it make sense? Right? Yes? No? Are you sleeping? Then wake up. Okay. These are examples, and I'm going to talk about each one separately when we start talking about the application. But briefly, if we talk about phase 1 metabolism or enzymes, you have the cytochrome P450, and the drugs that are involved or for which we have genetic testing or have been established to change or to influence drug response in CYP2D6, CYP2C9, C19, and others. Talking about phase 2 enzymes involved in the excretion and the elimination of drugs, you have 2 examples: transporters, another example, and then I will talk into more details about each one of them in next lectures. So, drug metabolizing enzymes, I don't know if you had the chance to think about the cytochrome P450. Cytochrome P450 has several enzymes, and each enzyme has several families, subfamilies, and isoforms. So, and do you know the difference? So we do have to establish the difference. I'm talking about the CYP2D6. Based on what's published and what's available, it looks like 10 to 20% of Caucasians in general are deficient in CYP2D6 gene, right? This gene is involved in the metabolism of so many drugs. So when we say 10 to 20%, we're talking about millions of patients who may not be expressing these genes, who may not be able to metabolize a drug. So when you don't metabolize a drug, if a drug is a prodrug, then you may not benefit from the active metabolite, right? And if the drug is an active drug and not metabolized, what's going to happen? It stays in your body for a longer time, doesn't get excreted quickly, so it may cause or create more damages in your system. Okay, so patients with no CYP2D6, they are either not getting the benefit, so they should not be put on a drug, they should be put on an alternative drug. Or if they are put on a drug that is not metabolized, they will be having some side effect that may be, you know, significant side effects if the dose does not change or if the drug does not change, and if the patient is not genetically tested prior prescribing the medication. This is another example about pharmacogenomics and drug transporters that you know, involved in the P glycoproteins that are resistant or are, you know, giving resistance to some drugs. So I'm not going to go into more details, but I'm going to have a chance to talk about each one of them into more detail. So right now, I'm going to move on to build some definitions. It is very important that you do know the exact definitions of, you know, or terminology for some words, and this is going to be part of the midterm. So I'm not expecting you to tell me 20-30%, I'm not going to quiz you on 20 or 30% Caucasians, but I am expecting you to know the exact definition. For example, what is the difference between genomics and genetics? What is the difference between the genotype? What is the difference between genetics? So, so I'm going to go, I know that you can read it on your own, but you may still, during the test, remember what I'm saying. So, what is a genome? What's in your genome? When someone says, what's in your genome? So you have to know, what is my genome? What is the definition of the genome, right? So it's all your genetic material. That is the genome, right? So genetics, evidence genetics is the study of one single gene, and genomics can be the study of all genes and their effects, you know, at one time. What is your genotype? So your genotype is defined by 2 genes because you inherit one from your mother, one from your father, right? So you have 2 genes contributing to a genotype, right? So when we talk about pharmacogenomics, I'm not going to talk about genetics in general. I'm going to focus on drugs, so, and on enzymes that are related to drugs. So I'm using again CYP2D6. So someone's genotype, someone in this room may have this CYP2D6 genotype, or will be determined by CYP2D6 star 1 and CYP2D6 star 4, meaning the patient or this individual have inherited one isoform from one parent and the other isoform from the other parents, right? So they have this individual may have inherited 2 different isoforms. So let's talk about biological variations. So you can have, some of them can be classified as neutral. What do we mean by neutral? Neutral, it means, you know, it's a variation that is due to the skin color, to the eye color, to the color of, you know, to your height, to your size, to your personality. So this is neutral. But you do have some advantages and disadvantages variations. If it's advantageous, meaning that it contributes to you, you know, being healthier, you have good genes, or to you, you know, being able to live in very cold environments, like people are, some people, you hear all the time, oh, I cannot stand, you know, hot, you know, or very warm environments. I cannot go to this country or this place, most likely because your genes have not been evolved to, you know, to stand for the hot environment or to stand for the cold environment. So these can be advantages to some patients or to some individuals living in under certain conditions and in certain environments. Disadvantages, when the genetic variation, for example, contributes to or leads to diseases or leads to non-responsible drugs. These are disadvantageous variations. So in pharmacogenomics, we are more interested in anything that is related and causing diseases, and maybe for you that is related to a drug response, because we're not talking about inherited diseases. They're not managed by, most of the time, by drugs, but we're talking about conditions that are managed by drugs. We're going to limit our focus to conditions that are really managed by drugs. Okay. So if you tell me that you remember what is a DNA, I'm going to skip this slide. Yes? Everyone? What is a nucleotide? Yes? Phosphate, sugar, and base. Okay, great. I'm going to skip this. What is the difference between RNA and DNA? One is double-stranded and the other one is, yes, one molecule. What is the difference? Another difference? Okay. So you feel comfortable skipping this information. It's going to be on your midterm. I don't have to explain it, right? I believe it was covered in
high school.
Human genetics and molecular biology. Okay, so now, most likely you know this information, maybe not, but our DNA is 3 billion base pairs. We are all identical, 99.9%, right? We have the same sequences. We are different by 0.1%, which is still 3 million bases. So our identity, and we are identical in a way that we have different hair color, we have different eye colors, we are different size, different heights, different personalities, different ways of talking, different ways of learning languages. But these are all mutual, making us who we are, different from each other. We have mutations that 0.1% differences that are advantageous or disadvantageous. So that makes our differences. But it's still 3 million things. And I'm not going to ask you about those numbers on the video. So, you know where is our DNA located, right? In the nucleus, and is packaged under chromosomes, right? You know what is a chromosome? Yes, no? Yes, sure? Okay. You know what do I have on this slide? Okay. This is a human chromosomes referred to as karyotypes. You have, each one of us here has 23 chromosomes, pair of chromosomes, right? That makes us identical in a way and different in a certain way. But what I'm going to point here is to a location. How do you know where a gene is located on your DNA or on your chromosome? Do you know how to read the location? Yes? No? No. Okay. This is going to be on the midterm because it's important. So if you think about your karyotype, it's a country, right? And the way we use the GPS, why do we use a GPS? To take you from one place to the other, to know where is the address located, right? Same thing with the karyotype. Think of it as a country and think of each chromosome as a town, right? And you want to find genes and where they are located, like their address. We are looking for an address for a gene. So let's use the cystic fibrosis gene as an example. Where is this gene located? We know where is the gene located, and it has been identified and established. And it's, I would say, I'm using just GPS as an example. So if you put the address, it will take you to the CFTR gene address or location. So let's start with the chromosome. So this is an example of chromosome. It has a little depression, centromere, and it has a short arm P and a longer arm Q, right? So every chromosome has 2 arms and depression, right? So based on technology and staining, we were able to identify where certain genes are located. We don't know everything yet, but we have good information about some genes and where they are located. So apparently, if the CFTR gene is located at position 7q31.2, we should be able to understand the meaning of this, right? So 7 means this gene is located on chromosome 7, right? Q means that the gene is located on the longer arm of chromosome 7, right? 3 means that if you look at this section over here, this is region 3. Okay, it has a different color, lighter color. So the CFTR gene is located on chromosome 7 on the Q arm within region 3, within band number 1, and the subband number 2. Right? So we know where the gene is located. If we know where the gene is located, if you know and if you hear about genetic therapy, and if you remember what I've said earlier, that people may want to go and change their genes, once you know the location, you can go to the location. And you can try to replace the defective gene with a more functional gene. So this is what scientists are trying to do, some in a good way, some in bad way, some in an ethical way. But we know that we can identify in the future, we will know everything about our genes and where they are located. So this is an example of where the gene is located. When you're going to receive a genetic test, or when you're going to receive information about patient genetic test, you may see something, you may come across something as the gene is at this position. So now you should be able to understand what is 7q31.2 mean. It's an address, takes you to where the gene is located, right? Does it make sense? Do you feel now confident you can read the location of a gene on a karyotype or on a chromosome? Right? Okay. So I think you already know about the gene structure. If you don't, I will go over it. I don't want to just, I don't want to be bored. Do you know the gene structure? Yes? Exons, introns, and translators. So let me go into something that I know you may know it, but I want to go into it because we're going to use it in future lectures. So you have 2 chromosomes, and you have 3 different genes. You have, and we want to talk about the genotype. Remember, the genotype is defined by 2 different genes that someone inherited from each parent. So if you talk about the first gene, or the first genotype, double P, upper P. So this individual is referred to as being homozygous for the dominant allele. Dominant allele means the allele that is highly functional and expressed. The gene next to it is an individual who is homozygous for this gene that has a gene, and the lowercase is known as a recessive allele. So this individual has inherited 2 recessive alleles, but he is homozygous because he inherited recessive alleles from both sides. The last one is dominant and one is recessive. So this individual is heterozygous because he has received different isoforms of the genes, right? So we're not talking about expression yet because these genes will be expressed differently based on being dominant and recessive. So remember, the genotype is determined by 2 different genes or isoforms that has been inherited from each parent. Okay. So let's talk about the gene variation. When you hear a wild type gene or wild type mutation, what does it mean in your mind? It's not mutated. It means that this is the reference mutation or gene. It means this is what most people have. It's like same normal functional gene. That's called as wild type. A variant gene, meaning that it has a mutation. For example, a G has been mutated into a T. So this sequence is a variant. So what does it mean having a single base or point mutation? Did you guys cover this in genetics?
Yes.
Okay, so I don't have to go. But you know, you may want to review it once we start talking about. Did you cover missense, nonsense, insertion, deletion?
Yes.
And
Yes. We don't need to take this class anymore.
It's been talking about nonsense the entire
They will
time.
give you your picture. So this is just an example of missense mutation where a change in one base leads to a change in an amino acids and may lead to a change in the function of a protein. Right? A nonsense mutation means that you have one change in this change was not aligned.
That's crazy.
A single nucleotide has been replaced and led to a stop codon. The stop codon can truncate the protein, and you're going to have a truncated protein that may not be functional. So this can lead, for example, to an enzyme that may not be functional, that is involved in the metabolism of drug. Insertion or deletions, meaning within your gene, you can see an insertion of a certain number of nucleotide, and they may be shorter, longer, but they may also cause a change in the way your protein or enzyme is responding. Something deletion, like part of the gene has been deleted and may lead to a non-functional enzyme. Okay, so polymorphism. We refer to polymorphism when mutation occurs in 1% or more in a population, meaning it's significant. So it's not only a variation or mutation, it's referred to as a polymorphism. And the best polymorphisms are, we know as of today related to a drug response or a variation in drug response is SNP, a single nucleotide polymorphism. There's a change in one nucleotide can lead in change in how the enzyme or the protein responds to the drug. So, and it can be also neutral because every 300 to 1,000 days you're gonna have a SNP, but SNP will not be changing anything. The SNP becomes important when it is to a genetic variation and polymorphism and variation in drug response. So. Short tandem repeat, was it covered? Yes? Okay. So let's talk about something that is related to drug response. So based on polymorphisms, patients have been stratified into 3 different metabolizers phenotype, right? A poor metabolizer, intermediate metabolizer, extensive metabolizer, and ultra-rapid metabolizer. In general, the extensive metabolizer is the normal, the standard metabolizer phenotype. Poor metabolizer is when the patient does not express the enzyme to metabolize the drug. Intermediate, if the protein function has been altered, so patient is metabolizing less of the drug, or the drug is metabolized less by the enzyme. Intra-rapid, when a patient has more copies of the gene, so more metabolism is happening, And you know what happened when you metabolize the genes quickly? So the drug quickly, so the drug does not spend enough time in your body to bring about the right response. So you cannot benefit from a drug when you are an ultra-rapid metabolizer, but depending on whether the drug is pro-drug or a drug. Okay. So let's go through some nomenclature and how to read the enzymes. So this is an example of human CYP2D6. Of course, human is the species because you may also come across CYP2D6 that is expressed in the rats, in baboon, in other species. So CYP is in reference to the superfamily. 2 is in reference to the family of CYP. D is in reference to the subfamily. And 6 is in reference to the isoform, right? Because you have CYP2D6, you have CYP2D4, you have CYP. So you need to know exactly which enzyme metabolizes the drug. So you need to be able to understand the terminology. So apparently CYP2D6 is located on chromosome 22q13.2. So what does it mean? So the location of this chromosome of this gene is on chromosome number 22, right? On the Q arm, the longer arm. What is 1? Is the region. What is a 3? Band. What is 2? Subband. So if you remember what we discussed earlier about the cystic fibrosis gene, is on chromosome 7. Right? This one is on chromosome 22, right? So these two genes may not be inherited together. May or may not be inherited together. 2 different chromosomes, right? So, CYP2D6 identifies the gene, right? And in general, when you talk about the genes are always, noted in italics, when you read papers or textbooks, and the protein is, in normal space. And let's take a look at the alleles or the variants. So, and what you have here, you have examples: the CYP2D6 star 1, star 4, and star 10. You have 3 different alleles, and we refer to this nomenclature as the star nomenclature. Do you know how to read this? Have you gone through the star nomenclature in genetics? Yes? No? Okay. So what does it mean? It means that, someone may be expressing CYP2D6 star 1, another one CYP2D6 star 4, another one CYP2D6 star 10. So different alleles. We have more than 3 alleles, when we talk about the CYP2D6 enzyme. We have identified almost 100 alleles, so in the population, people or patients or individuals may be expressing different enzymes. So we have to understand whether they are expressing the functional one, the standard reference, wild type, or another variant. So the genetic test most likely will come with CYP2D6 star 1 or star 4 or star 10, so you do understand what does it mean, okay? But it also may come as CYP2D6, 1846 G2A, or 100C into a T. So what does it mean? So you should be able to understand the meaning of, these isoforms or these variants. So the first number, if you talk about 1846, meaning that CYP2D6, meaning at position 1846 nucleotide, the G is positioned. If you think about the gene has a sequence. It may be 100, 5,000, or 6,000, or even more nucleotide, right? Assuming this CYP2D6 has 3,000 nucleotides, right? If you go from 1 to 3,000, G is on position 1846, right? And the C is at position 100. It's the 100 nucleotide, is a C. This G is at position 1846. And why we are talking about this G and this C? Because they have mutated. G has changed into an A, and C has changed into a T. So this is a different variant. So we know which position the mutation is located, and we have to understand, how we can refer to it and at what position. So apparently, GS and the C is the wild type, the standard type, the functional type, or the functional variant. The A and the T are just another variant. So you can refer to a gene, you can have a genetic test in reference to CYP2D6*1 or in reference to CYP2D6 with a mutant or variant, talking about where the nucleotide variant took place. Or the genetic test will just come with a reference SNP, because a change in the nucleotide is a single nucleotide polymorphism, is a SNP. So this is a reference SNP, right? And why it's important? Because when you start looking at genetic testing, you have to understand, this is how you have to read the genetic test. In order for you to read the genetic test and interpret the test, you need to make sense of the information, or the information has to make sense to you, so you have to understand it. So at the end, CYP2D6*4 or CYP2D6 at this variant with a change of G2A at this position, or this reference number, they all refer to the same allele. But guess what? You have several genetic tests available. Some of them will come as CYP2D6*4. Some of them will come as reference. Some of them will come by position. But at the end, you should be able to read all of them and understand all of them and do the interpretation for every single test. So again, example of a genotype is CYP2D6*1*4, and the second genotype is CYP2D6*4*4. So we have two patients with two different genotypes. What does it mean? Then I receive this information into genetic test, then you have to draw a conclusion. Most likely the conclusion will be within the test, but you will have to explain it to your patient. Okay. So we're gonna review this table again, but just quickly, CYP2D6*1 refers to the extensive metabolizer. It's the standard. Most people have this metabolizer phenotype. It looks like 32% of Caucasians and 43% of Japanese do express the wild type. If you look at the CYP2D6*4, it's a poor metabolizer phenotype. It looks like 17% of Caucasians and 0.2% of Japanese, express or are poor metabolizers. But 17% of Caucasians is a lot. It's millions of people who are not expressing CYP2D6. If we talk about CYP2D6*10, it's an intermediate metabolizer, right? So this population, and if you look at the Japanese, 38% are intermediate metabolizers compared to Caucasians, where it's very low frequency. So you don't only need to know the variant or the mutant, you need to know how the variant and the mutant affects an ethnic group or affects population, so that if you look at the patient's family information, or you ask patients most of the time, now there is a questionnaire also says, please tell us about your ethnicity or your race or whatever, because it is helpful in this case, in pharmacogenomics, to know which population or which ethnic groups you belong to. For example, if someone knows that if you say Caucasian, they say, well, only 17%, or only 32% express the wild type. I don't know if this patient is a poor metabolizer. I don't know if this patient belongs to the 17% of Caucasians who are poor metabolizers, so maybe it's worth to prescribe genetic test to identify the genotype for this patient so that we can prescribe the right drug and make a right recommendation. The C into a T, yes, it means that on C to the 6 star 10, the C located at position 100, if you look at the start of the sequence of the gene, at position 100 in the wild type, there is a C. But for an intermediate metabolizer, the C has been changed into a T. That's why it is referred to as a star 10. But in the extensive metabolizer, at position 100, you have a C. Okay? So now you know that one single nucleotide change can affect drug response. So it doesn't have to be a whole sequence that has been deleted or added, or like one SNP can make a difference. So it's important to know that it is this SNP, because on CYP2D6 you may have hundreds of SNPs, right? But they may not all be involved in changing drug response. They may be neutral, right? No effect. But this one is making this allele to be an intermediate, or the patient to be an intermediate metabolizer. And this one, CYP2D6 star 4, change of gene to an A, is making this allele deficient and the patient a poor metabolizer. Does it make sense? Yes, yes. Star 1, star 4 are different alleles. Yes. It's not different chromosomes. Let me make corrections here. If you remember, it was chromosome. Everyone here, everyone worldwide, is expressing CYP2D6 on chromosome 22, not on a different one, right? So we all have this gene on our chromosome 22. When it comes to the sequence of the gene, we all have different variants or isoforms. Remember, every 300 to 1,000 nucleotides, we have a SNP. Most SNPs are fine, are not causing anything. But some of them, one of them, may make a difference, because one of them may be involved in how the protein is on how the gene is translated into a protein, and how the protein is folding into its first, secondary, or tertiary, structure. So, for example, for CYP2D6 star 4, a change of a G into an A at position 1846 has led to a poor metabolizer phenotype. This patient does not express CYP2D6 star 4, has no enzyme activity. If the patient is on a drug that requires a metabolism from a poor drug to an active drug, the patient cannot do it. So the patient is taking a drug that is not beneficial to him or her. So this patient should not be on a drug that is metabolized by CYP2D6. Right? So, and why the location is important? Because you may have a change of the gene to an A at location of 1500. But that location is not important, because that change from gene to an A at 1500 did not affect the protein function, did not contribute to a metabolizer phenotype. So that's why it's important to know that it's this position. And don't worry, I'm not going to test you on the position. I'm going to test you on your understanding of the information. I'm not going to, tell me which 1864 or which 100 yet. Yes. This is how you know to change when you read the. Okay, it's not great. No, this is just how you will understand and read the information. It is a change. There is a nucleotide that is guanine at position 1846. That nucleotide has mutated on the same position or at the same position from a gene to an A, right? So you go back.
What was the was it carbon?
So if I use this as an example,
No, carbon was the second class.
for example, so if I use this slide as an example, assuming that this A is at position 100. So this A has changed into a C at position 100. It's the same position, right? So when it changed from A into a C, that this enzyme is no longer functional. So this enzyme will contribute to a poor metabolizer phenotype. But if the A changes, if at this position, assuming this is position number 5, A changes from A, this is A to A. Okay, I cannot. This is the only example here. So if A, for example here, if A, assuming that A has changed from A to a T, but the change did not change the amino acid, so does not affect the enzyme function. Right? So you have to know which A has changed. Is it the one at position 2? Is it the one at position 4? Is it the one at position 100? To know which one affects the function of the protein.
What was the
So everything here is for you to understand how to read the information, so that you can make an interpretation of the test and what happens after you make that interpretation.
Separate.
But we're going to go over several examples that we can
It's not a pharmaceutical spy?
Same.
Okay, so I'm going to
Same like
longer
pharmaceutical.
than this, but we'll go